Hello. Chapter 4 it's about the functional anatomy of prokaryotic and eukaryotic cells. So in this chapter, we're going to learn what is the difference between prokaryotes and eukaryotes, what are their individual characteristics, and we also going to identify different types of bacteria, their arrangement, their names, and also we're going to learn the structures and function of the bacterial external structures, the cell wall, the internal structures, and we're going to see similarly to that the same for the eukaryotic cells, along with what are the mechanisms for moving materials across the membranes. So just to have a little introduction about what prokaryote and eukaryote means. So prokaryote, it's a word that comes from the Greek roots pro, which means pre or before, and karyos, which means nucleus. So basically, what this word means is before nucleus or pre-nucleus. Eukaryotes come from the root of the Greek words which are non-eu, which means true, and karyos, which means nucleus. So in essence, this word eukaryote means true nucleus. So just as a two-column comparison between eukaryotes and prokaryotes. So prokaryotes are cells similar to eukaryotes, but prokaryotes, they have in their cells typically one circular chromosome, and that circular chromosome is not found in a nucleus, with the exception of one bacteria is called Gemara obscuroglobus, which has a membrane that encircles the DNA. And then we have other bacteria that instead of having one circular DNA, they might have linear DNA, or they might have two chromosomes instead of one. But those are the exceptions. But for the most part, prokaryotes have one circular chromosome, and it is not membrane bound, so it's not found in the nucleus. Now, eukaryotes, they do have nuclear membrane, and they have paired chromosomes. The number of chromosomes depends on the cell type of the eukaryotes, but at least for human cells, we have 46 pairs of chromosomes. And then the DNA in prokaryote is not wrapped around proteins that we know as histones, while in eukaryotes, the DNA wraps around to be packed in proteins that we know as histones. Now, prokaryotic cells, they don't have organelles, and eukaryotic cells, they have membrane-bound organelles like mitochondria, like endoplasmic reticulum, Golgi complex, etc. Now, bacteria, they have a cell wall, and the cell wall is made out of peptidoglycan. There is one exception in which one bacteria doesn't have a cell wall, which is mycoplasma. But the rest, they have a cell wall that is made out of complex known as peptidoglycan. Now, eukaryotes, they have some of them, eukaryotic cells, some of them, they have a cell wall, not all. Animal cells, they don't have a cell wall. But some of them, they have a cell wall, and the cell wall is made out of polysaccharides. Archaea, which is a prokaryote, have cell wall, but the cell wall is not made out of peptidoglycan. It's made out of a component that we know as pseudomurine. And in terms of division of cells, prokaryotes divides by binary fission, meaning that one cell splits into two. And then eukaryotes, they divide either by mitosis, the cells, or by meiosis. And animal cells, as I said before, they don't have a cell wall. So this is the structure of the prokaryotic cells. So they have this cell wall, which is made of peptidoglycan, and then they have a plasma membrane, and they have a cytoplasm with ribosomes, and instead of having a nucleus, they have a nucleoid. And anything outside of the cell wall, it is considered an external structure of the bacterial cell, while anything inside the cell wall is an internal structure. Now, some bacteria, aside from having the DNA found in this region of the cytoplasm that we know as the nucleoid, they will have an extra piece of DNA. It's circular DNA, but it's smaller than this nucleoid, and this is known as plasmid. Now, some of the external structures will be pilus, fimbriae, flagella, while some of the internal structures will be inclusions, ribosomes, nucleoid, and plasmid DNA. And of course, we have the plasma membrane inside the cell wall. So now let's talk about the shapes of the prokaryotic cells. So the cells, prokaryotic cells, they have an average size of between 0.2 to 2 micrometers in diameter times 2 to the 8 micrometers in length. Most of the bacteria, they have a single shape. They are called monomorphic. And few of them, like rhizobium, they can have more than one shape. These are pleomorphic. Now, the shape will tell you what will be the name for the bacteria. So if the bacteria has a rod shape, it will be called bacillus. If the bacteria has a spherical shape, it will be coccus. If the bacteria has an spherical or helical shape, it can be called Aspergillum, Vibrio, or Spirochete. Other shapes can be like stars, or they can be rectangular, or they can be a combination of spheres and rods. So this will be called coccobacillus. So this is a diagram to show you the different shapes and also how the bacteria arrange or form groups. So all of these that you see in this left column are spheres. So all of them are cocci. Cocci is plural for coccus. Coccus is singular. And all that you see here on the right side column are all rods. Now, cocci, when they divide, they can divide either in one plane and they can form groups of two. And if they only remain in groups of two, are called diplococci. Diplo means two. Now, they can keep dividing in one single plane, and they do not separate. But instead of forming diplococci, they form a chain of cocci that we know as streptococci, like you can see here. So all of these images are scanning electron micrographs. So these are really nice magnifications where we can see the details. So these are streptococci then, and then if they divide in two planes, like in this plane and in this plane, you form a group of of four, and in this case you call it a tetrad. So we have 1, 2, 3, 4. Tetra means four. Now, if you divide in two planes, like here, here, and then horizontally, you will end up with a group of 8 cocci that we know as sarcinae, which is this. And then some of them, they divide in multiple planes like sarcinae, but they remain attached. And they form clusters that looks like grapes. And this will be called staphylococci. Staphylo actually means in Greek grapes. Now, bacillus, they can be forming only single cells that we know single bacillus. And if they divide and they don't separate and they form groups of two, you call it diplobacillus. Now, this division can continue in some bacillus, but instead of forming groups of two, they form a chain, and this will be called streptobacilli. Now, some bacteria, like bacillus or like cocci, they can have a shape that it is different in their ending. Like the ending typically of the bacilli, it's kind of curved, but sometimes it can be rounded, and they can be short, the bacillus. So it's kind of intermediate form between a coccus, like an spherical shape, and a bacillus. So in this case, you would call it coccobacillus. Now for the spiral bacteria, if they are short, single, and they are coma-shaped, this will be called vibrio. So as you can see here in this scanning electron micrograph, they have a shape of a coma. And now, if they're short and thick, but they have the shape of a spiral, this will be called spirillum. And sometimes if they're thin, slender, and they're long, this will be called spirochetes, like in this case. So all of these are spiral bacteria. They look like a helix, at least for these last two, the spirillum and spirochete. But this one is thicker and shorter, and this one is longer and thinner. And the vibrio is short, and it has a coma shape. Now, in microbiology, we have this name bacillus, but this bacillus can be referring to the shape of the bacteria, or it can be referred to the species or the scientific name. So in the case of the scientific names of the genus of the bacteria, remember, you always use capital letter in the first letter of the name, and then you use italics. So in this case, the scientific name will be bacillus. And the bacterial shape of bacillus is actually a rod. So in this case, you will use only lowercase and non-italic letters or font. So this is then how bacillus look like. They can form a long chain of rods, and they can form this tangling type of bacteria. This is a light microscopy. Anytime that you see these squares with these letters, that means the type of microscope that they use. And this is the size scale or size bar. So then now let's talk about the characteristics and the names of the bacterial structures, external structures. So glycocalyx, it is an external coat of the bacteria that usually is sticky. And this glycocalyx is made of polysaccharide. Polysaccharide is basically a polymer of saccharides or sugars. And this extracellular material, the bacteria uses it for attaching into surfaces to form biofilms. So this is an example of a coating of bacteria made out of glycocalyx. Now, glycocalyx can be subdivided into two different types. It can be a capsule if the glycocalyx is neatly organized and compactly packed, the polysaccharides, or it can be a slime layer if the glycocalyx is unorganized and loosely connected. Now, the function of the capsules, the organized glycocalyx, is to prevent phagocytosis. So this bacteria, which is called Streptococcus pneumoniae, it's a diplococcus. It has two cocci. This one has a capsule. So this bacteria causes pneumococcal pneumonia, and this bacteria it's very hard to get rid of because it has this capsule that prevents the cells of the immune system to attack it. So anytime that a person has an infection with an encapsulated bacteria, this is a serious infection. Another type of external structures will be flagella and axial filaments. So flagella are long extensions that helps the bacteria to move into what we call runs or tumbles. Runs is basically a movement that is continuous that goes towards a particular direction. And tumbles are short movements that are in random direction that helps to the bacteria to switch, to go into a different direction. Now, within the flagella, there is this protein that is called flagellin. And these flagellar proteins can be identified in certain tests in microbiology, helping us to identify what we call zero-bars or subspecies of bacteria. Like, for instance, in E. coli O157H7. H7 refers to the flagellin protein in the flagella of E. coli, and this is known as H antigen. So this bacteria causes severe diarrhea. Serious diarrhea is one of these emerging infectious diseases, E. coli O157H7. Now, the flagella can be of different types. So if the bacteria doesn't have flagella, it's called atrichose. If the bacteria only has one flagella, it will be called monotrichose. Mono means one, trichos means hair. Iophotrychus would be a bacteria that has, instead of one flagella, has a tuft. And Amphitrichus will be when the bacteria has flagella on both ends of the bacterial cell. Polar will be when it's found in only one end, and Peritrichus will be when the bacteria has flagella in the entire surface of the cell. Now, there are some bacteria that moves, but they don't use flagella. Instead of using flagella, they use axial filaments, which it is also known as endoflagella. Endo means inside, flagella means flagella, like this long extension. But instead of being externally located, the axial filaments are located inside the cells, and they're made of fibrils, like proteins, that are found beneath the outer sheet of spirochetes. And they help the bacteria to move in a spiral or corkscrew motion. So this is a diagram to show you the types of flagella. So this bacteria, it's a rod, and it has this long flagella that is found all over the surface of the cell. So this will be peritrichous. Peri means around. And then this one is monotrichous and polar. Why? Because it only has one flagella, as you can see here, and it is found on one end. Now, this one, it has a tuft, so it has 1, 2, 3, 4, 5, 6, or 7 flagella, and it is found in one end. So when it's found in the ends, it is called polar. And in this case, it's amphitrichose. Amphi means two. And this is a spiral bacteria that has this flagella in here and this flagella in here. So it's amphitrichous and polar because it's found in the ends and it is not found all around, like in that case. So details of the flagellar structure. So the flagella has this long extension that it is outside of this wall of the bacteria. This is the filament. And the filament, it is attached to the hook. The hook is located next to this external cell wall of the bacteria. And this hook is motile, so it can rotate. So this is gram-negative bacteria cell wall, and then gram-positive bacteria cell wall. So the hook itself, it is connected to this basal body. And the basal body has this cylindrical disc-shaped structures that embed within the plasma membrane, in the case of gram-negative bacteria, to the outer plasma membrane, which is this, and the inner plasma membrane, which is this. So here we have 4 discs for this basal body of this flagella of gram-negative. And then you have 2 discs in the basal body of this gram-positive bacteria that has only one plasma membrane. And this is the type of movement that the flagella can make. So the flagella can rotate counterclockwise or clockwise. And with this, they can move towards a surface if there are food available, or away from that surface if there is a chemical that damages this bacteria. And then when you have a continuous motion towards a specific direction, like in this case, or in this case, or in this case, you call it a run. And then you are rotating your flagella. And if you want to make a tumble, you stop the rotation and then you redirect the bacteria. So tumbles are more momentaneous movement and runs are more directed and more continuous. And then some bacteria, like in the case of proteus, this is proteus, they have multiple flagella. It's peritrichous flagella. They have more than 1,000 around. So they have a movement that is called swarming. Axial filaments are located, as I told you, inside the cell. So this is the protein inside this spirochete that is known as leptospira, this one in axial filament shown here. And if you magnify, they're located inside this outer sheet that protects these spirochetes. So when this contracts, this endoflagella, the spirochetes, they perform this twisting motion that looks like a corkscrew. Other external structures will be fimbriae, which are hair-like short appendages that are made of peeling protein. And these fimbriae help for the bacteria to adhere to surfaces so that they can form a biofilm. Pili are longer than fimbriae, and they have a thinner diameter than flagella. And this pili helps for the bacteria, in some cases, to attach, but also they help for the bacteria to perform gliding or twitching motility or to transfer pieces of DNA. So this is a rod, or actually a diplobacillus, that has multiple pili, and they have multiple fimbriae. So the longer extensions will be the pili, and the shorter extensions will be the fimbriae. And plasmid DNA will be transferred with this pilus. So plasmid DNA helps some bacteria to survive the attack of antibiotics. So those are the external structures of bacterial cells. Now, let's talk about the characteristics of the cell wall of bacteria. So the cell wall of bacteria is made of complex of proteins and saccharides that we know as peptidoglycan. Peptidoglycan is made out of rows of disaccharide polymers, and these disaccharide polymers are made of two carbohydrates, N-acetylglucosamine or NAG and N-acetylmuramic acid or NAM. And this NAM, it has attached a link of tetrapeptides or 4 amino acid proteins, and the adjacent rows of these NAM and NAG are held together by polypeptide chains, and they form a cross bridge. What are the functions of the cell wall? It prevents the bacteria to burst because it might be subject to hypertonic environments. We will see in just a few slides what is hypertonic, but the bacteria can burst under the wrong conditions. And then the bacteria will maintain its shape as well because these cell walls are semi-rigid, and some of the components of the cell wall help to identify bacteria. Now, this is the structural components then of the peptidoglycan. So this is N-acetylglucosamine, NAG, which is similar. It's a sugar, it's a carbohydrate just like this, N-acetylmyrmic acid, but look at the difference. So this one has a smaller side chain in here than this one. This one has a longer side chain. And then we have tetrapeptides attached to this NAM, which is this. So here is basically a representation of these polymers of NAM here in pink color and NAG in brown color. So that's the alternate arrangement, NAM, NAG, NAM, NAG. And attached to this NAM, we have these tetrapeptides, these 4 amino acids, and then we have these polypeptides that helps to make sure that these rows don't separate. So these are the carbohydrate backbone held by these tetrapeptide chains and these peptide cross bridges, which are these. Now, depending on the type of bacteria that you are talking about, it is how many layers of peptidoglycan you will have. If you have a gram-positive bacteria, like in this case, this is a gram-positive. If you magnify a piece of this peptidoglycan shown here in brown color and the plasma membrane shown here in yellow color, if you magnify this, you will see that the peptidoglycan in gram-positive bacteria, it is of several rows, like 25 to 30 rows. And then if you magnify a little piece of this gram-negative cell wall, this one in brown color, you can see that the cell wall is actually sandwiched by the outer membrane here in the top yellow and the inner membrane in the lower yellow. So if you magnify this and if you see it, you have 1 or 2 rows of peptidoglycan. So peptidoglycan is less abundant in gram-negative bacteria than in gram-positive bacteria. And then if you see a scanning electron micrograph of bacteria, you can see that this bacteria has this dark line underneath the capsule. So this is an encapsulated bacteria. So this is the dark line that represents the cell wall. So the cell wall is dense. And then here you have the cytoplasm of the bacteria, ribosomes inside the cell wall, and then you have the nucleoid and inclusions. Now, gram staining technique helps to distinguish between gram-positive and gram-negative bacteria because of the differences in the cell wall. So when you add the first dye in gram stain, which is crystal violet will enter and penetrate both gram-positive and gram-negative bacteria. But when you add this iodide, which is the mordant, this crystal violet will form complexes with this iodide. So they will form crystal violet iodide complex, CVI. And in the next step, when you add the alcohol will dehydrate the peptidoglycan of gram-positive bacteria. And because these crystals of crystal violet and iodide are big, they won't leave the cells of gram-positive bacteria. And remember, since crystal violet stains purple the cytoplasm of bacteria, the gram-positive bacteria will remain purple because it will have entrapped these crystal violet iodide complexes. Now, gram-negative, they have a very thin peptidoglycan, and they have the outer membrane. And alcohol, it dissolves the lipids of the outer membrane and will make holes in the outer membrane and in the peptidoglycan. So once you rinse the alcohol from these gram-negative bacteria, the crystal violet iodine complexes will wash out. And then these bacteria at this step will look colorless unless you add safranin, which is the counter stain for gram-negative. So this is then the differences between the gram-positive and gram-negative cell walls. So gram-positive cell walls, they have a thick peptidoglycan layer, like 25 to 30 rows or layers, sorry, while this one gram-negative has like one row. Along with that, the gram-positives, they have in their wall this component that is known as teichoic acids that helps to identify gram-positives. And if the gram-positive, they have a flagella, they will have two rings or two discs in the basal body of the flagella. While the gram-negatives, they have another membrane, one layer of peptidoglycan, and four rings or discs in the basal body of the flagella, if they do have a flagella. Now, the wall of the gram-positives can be broken down by the following, by this enzyme that is known as lysozyme, and also by this antibiotic, which is known as penicillin. Now, the gram-negative cell wall has components that creates toxicity in the host. So these are known as endotoxins. And the gram-negative cell wall, it will be damaged by this antibiotic, which is known as tetracycline. Now, the gram-negative cell wall, it is sandwiched between the outer membrane and the inner membrane, or the plasma membrane of gram-negatives. And that space where you have this cell wall is known as periplasmic space. Now, the cell wall of gram-positives, they have polysaccharides that helps to identify the bacteria and classify them into different groups, especially if it's one type of bacteria that we know as Streptococcus. So this is just a summary of the characteristics of gram-positive and gram-negative bacteria. So remember, gram-positive stain purple, while gram-negatives stain red. These ones retain crystal violet. These ones do not retain crystal violet, so they need to be counterstained with safranin, and they can stain pink or red. Now, some bacteria have what we call atypical cell walls. And this means that they don't have same components as the cell walls of other bacteria, or they might not have a cell wall at all. Examples will be acid-fast bacteria. So acid-fast bacteria has acid-fast cell walls. So they have outside the peptidoglycan a waxy coat that is known as mycolic acid in the case of mycobacterium. And this will prevent from the normal dyes that you use in gram staining to penetrate into the cytoplasm. So in this case, you have to use another technique to stain them, which is known as the acid-fast staining. So in this case, you use a dye that is called carbolfuchsin, that it will stain red the bacteria that resist the decolorization step with acid alcohol. And in order to open these waxy lipids in the cell wall of acid-fast, you need to steam the bottom of the glass slide where you have the bacteria so that you can open up with the heat these mycolic acids so that the carbolfuchsin can penetrate the cytoplasm. And once the carbolfuchsin penetrates the cytoplasm, it will stain the cytoplasm, and then you add acid alcohol, and then you rinse, and then you add your counter stain, which is going to be methylene blue. Now, if the bacteria is acid-fast, then it won't decolorize after you add carbolfuchsin, which is red. But if it's non-acid-fast, it will decolorize, and then it will pick up the counter stain, which is methylene blue. And then acid-fast, then positive cells will stain red, like in this case, and the ones that are non-acid fast will stain blue, like in this case. So these are non-acid fast bacteria, and this is acid fast bacteria. And then along with Mycobacterium, we have Nocardia, which also is an acid fast bacteria. Now, some bacteria, they don't have a cell wall, like in the case of Mycoplasma. Mycoplasma is a very small bacteria. Instead of having a cell wall, it has a plasma membrane. And in the plasma membrane has these lipids that are known as esterols that makes this plasma membrane to be a little bit rigid. Now, archaea would be prokaryotes that either don't have a wall, so they will be wallless, or if they have a wall, instead of having peptidoglycan, they have this chemical that is known as pseudomurine. That is similar to NAM, but it's not NAM. And they don't have this type of amino acids, the amino acids, in their cell wall. Bacteria, they have the amino acids and L amino acids, but these archaea, they have only L amino acids, and they don't have NAM, or N-acetylmuramic acid. Now, one of the important points about knowing these characteristics of bacteria is that since bacteria, they don't have the same type of components in their structures, including the cell wall, as other cells like animal cells, these differences can be used to treat bacterial infections. So you can destroy the components of the bacterial cell walls or the bacteria without damaging the host cells. So that's the important point. Now, how can you damage the cell wall of bacteria? Well, naturally in our body, we produce this enzyme that is called lysozyme that is found, for instance, in tears, saliva, or in secretions of the digestive system. And this lysozyme enzyme will break down the links between NAM and NAG. So it breaks down the disaccharides in the peptidoglycan. Another way that we can damage the cell wall of bacteria is by using antibiotics like penicillin. Penicillin prevents the formation of peptide bridges in peptidoglycan. And when you have then this damage to the cell wall, the bacteria can either don't form a cell wall, and in this case will be called proplast, and if it's a gram-positive, you would call it an spheroplast. Now, when you have a weak cell wall, when you have weakened the cell wall, these proplast or spheroplast can burst because they are susceptible to osmotic lysis. Other bacteria, they can take they can swell into irregular shapes, and they will be called L forms. But not because they take an L shape, it's just because they were discovered in the Lister Institute. So those are the characteristics of the cell wall. Now let's talk about structures internal to the cell wall, like the plasma membrane. So the plasma membrane is made of phospholipid bilayer, peripheral proteins, integral proteins, transmembrane proteins. So this is the cell wall in brown. And at least for this bacteria, which is a gram-negative, you have an internal plasma membrane and an external plasma membrane that you can see here in this transmission microscope with these two rows, which is basically a double membrane. If you magnify this region, you will see the components. So it's made of two layers of lipids. Lipids, they have a phosphate head, which is this, which is polar, and glycerol, which is an alcohol. So this polarity of these components helps them to be able to interact with water inside the cells, which is this part, or outside the cells. Attached to these polar heads of phosphate group and glycerol, you have two fatty acids tails that face internally because they are hydrophobic. They don't interact with water. So then you have phosphate heads facing outside and inside the cells, while the lipids in the phospholipid bilayer, which will be the fatty acids, faces internally. And embedded within this lipid bilayer, you have these integral proteins that can be open, like in the case of channels or pores, or they can be just found expanding through these lipid bilayers and connected to internal peripheral proteins or to sugars and proteins outside the cells to make the cells to detect signals in the environment. Function of the plasma membrane to maintain selective permeability. It allows the passage of some molecules but prevents the passage of others. In the prokaryotes, there will have plasma membrane enzymes to produce ATP because prokaryotes, they don't have mitochondria to produce ATP. Other prokaryotes, they will have pigments in foldings that are known as chromatophores or thylakoids that allows them to undergo photosynthesis. So in this case, this is a bacteria, this is a long rod, and you have here the chromatophores that helps this bacteria to produce photosynthesis. Now, in the case of gram-negatives that has another membrane outside of this peptidoglycan, they have also an inner membrane. And the space between the inner and the outer membrane is known as periplasmic space. This space is very important for this bacteria because it will have enzymes in here that helps to provide this bacteria to survive the attack of cells of the immune system that is known as phagocytes, or attack of proteins in the body that are known as complement system, and also prevents the destruction by antibiotics. So periplasmic space is a mechanism of defense for gram-negatives. Now, in the outer membrane, the gram-negatives, they have a lipid that is known as LPS, lipopolysaccharide. And lipopolysaccharide has an O polysaccharide that it has this component, that it is an antigen that helps you distinguish gram-negatives, like in the case of E. coli O157H7. O stands for O antigen found in the lipopolysaccharides, and H7 is for the flagellar protein or antigen. Now, the lipid portion of this LPS is called lipid A, and this is the endotoxin. So this, when it's released, it causes fever, chills, and sometimes increases the body temperature when someone has a gram-negative bacteria infection. And then within the plasma membrane, this outer membrane, the gram-negative has proteins that are known as porins, that are channels that helps for import of nutrients and extrusion of wastes by these gram-negatives. Characteristics of the plasma membrane of gram-positives is that they have only one lipid bilayer with protein channels, and they have plasmic space, which will be the space between the cell wall and the plasma membrane. And in this plasmic space, the gram-positives, they have lipoteichoic acids. Now, the gram-positive also have teichoic acids, but the teichoic acids will be attached to the cell wall, while the lipoteichoic acids are attached to the plasma membrane. And these teichoic acids will be useful to identify different types of gram-positives. So just to see then the differences between the cell wall and the proteins, and the plasma membrane of gram-positive and gram-negative bacteria. So if you magnify this area from the gram-positive cell wall and the plasma membrane, this is what you will see. So in here, in this plasma membrane of this gram-positive, you can see this long extension. This is the lipoteichoic acid. that connects the plasma membrane to the cell wall. So this is the cell wall. And then within the cell wall of gram-positive, you have these teichoic acids. So these will be useful to identify then certain types of gram-positive bacteria. Now, if you magnify then this region of the wall and the membranes of the gram-negative, this is what you will see. So you have the inner membrane that is called a plasma membrane and the outer membrane. And in between you have the single layer of peptidoglycan. And what is the name of this space between the outer membrane and the inner membrane? Periplasmic space. What does this bacteria has in the periplasmic space? Enzymes, protective enzymes. And then here within this plasma membrane, it has integral proteins, and of course it will have channels that allows for the passage of nutrients. And in here also the bacteria in the outer membrane will have as well these integral proteins, but also will have enzymes that allows these bacteria to break down large nutrients and to bring them in to the cells. And then gram-negatives, they also have these LPS or lipopolysaccharides. All of these in blue color that you see here are LPS. So if you magnify one of these LPS, this is what you will see. So you will see this lipid A that it is attached to this outer face of the plasma membrane, which is this. And then you have a core polysaccharide that is attached to the lipid A. And then at the tip of this LPS, you have the O polysaccharide. So this tiny tip that you see here is the antigenic, the one that allows to distinguish bacteria. So for E. coli O157H7, this little tip is where you have the difference that makes this bacteria to be more aggressive. And this lipid A is the one that causes fever, chills, and muscle aches, the signs and symptoms of gram-negative bacterial infection. Now, the plasma membrane in bacteria can be damaged by alcohols, detergents like quaternary ammonium, and antibiotics like polymyxin. When you create pores then within the plasma membrane, you will have something that's called plasmolysis, in which the cytoplasm leaks through this plasma membrane. Now let's talk about the movement of materials across the membranes. So materials can move from an area of high concentration to low concentration, or from more to less, into what we call simple diffusion. So the molecules just cross through this plasma membrane. So here you have more outside and you have less inside. So these molecules will move from more to less until they reach equilibrium. And this is the demonstration that we typically do in the labs. So you have a beaker filled with water, you add a crystal colored molecule, and then the particles of these molecules will diffuse from the bottom of the beaker towards the sides and up in the water until all the water will be colored of the same intensity. Now you can have as well the movement of particles from more to less, but now using these transporters or channels of protein within the plasma membrane, and this will be called facilitated diffusion. So facilitated diffusion can happen through a non-specific transporter in which the molecules that are small enough and they have the right charge, in this case positive charge, can go through, or they can physically connect these particles into the outer part of this transporter, and then the transporter will pass this molecule and release it to the inside of the cell. Like in this case, this is a glucose transporter. So this is specific for glucose and not for other molecules. Now, water also can move through this plasma membrane. And this movement happens through pores of water that we know as, well, pores for water that are known as aquaporins. And the movement of water across a selectively permeable membrane is known as osmosis. And water will move from an area of high concentration of water to low concentration of water, or from low concentration of solutes to high concentration of solids, so that it can dissolve these solutes. And once water moves, water will not move indefinitely. It will be stopped by a physical force that we know as osmotic pressure, which helps to prevent the movement of water across the membrane when it's not necessary. And this is as well one of the typical examples that is used to show the principle of osmosis. So in here you have a glass tube connected to this cellophane sac where you have a glucose solution, sucrose, 20%. And then you have this sac sealed in this area, and then you introduce this sac to a beaker with water, but no sucrose within this beaker and the water inside the beaker. So when you introduce initially your sac of cellophane, which is a semi-permeable membrane, it will allow only the entrance of water, but it won't allow the exit of these purple spheres, which represent sucrose. So sucrose won't exit, but water can enter or exit the cellophane. Now, since you will have no sucrose in here, you will have very poor osmolarity. So you will have a hypotonic solution, what we call a hypotonic solution, where you don't have so much solutes. So water will move then from an area of low concentration of solutes to high concentration of solutes, or from more water to less water. And you can see that with the arrows. So you will keep doing that, and then the level of the water in the beaker will decrease because it's entering into this cellophane sac, and then the water ultimately will rise through this glass tube until you reach a particular pressure that is known as osmotic pressure, and that will prevent the water from falling out from the glass tube because no more water will enter. And then you will have equal concentration of water inside the sac and outside the sac. So you won't have movement of water. Now, bacteria is found in the environment, and in the environment can find different situations. So bacteria has internally in their cytoplasm a particular concentration of solutes. And if these solutes that are found within the bacteria internally are equally concentrated as the ones that are found externally, that solution in which the bacteria is in, it will be called isotonic solution. So you don't have movement of water in and out. The bacteria won't gain volume, but it won't lose volume. Now, if you place a bacteria in a hypotonic solution in which you have less concentration of solids, and solids are represented by these small dots. So if you have this solution with small concentration of solids, hypotonic, hypo means low, tone represents how much solids. So if you have less solids outside of the cells than inside of the cells, then the water will move into the cells to dilute these solids, and then the bacteria will burst through osmotic lysis. It will gain volume, it will weaken the cell wall, the plasma membrane, and it will burst. Now, in a hypertonic solution in which you put, for instance, the bacteria in a solution where you have a lot of salt outside or a lot of sugar, the water inside the cytoplasm of the bacteria will move from high concentration of water to low concentration of water to dilute all of these solutes outside, and the bacteria will lose volume. So this will be dehydrating the bacteria, and it will move the plasma membrane away from the cell wall, and that is going to cause a death signal, and the bacteria will die into what we call plasmolysis. So then water will move from areas of high concentration water to low concentration water through these aquaporins, or these water channels in the plasma membrane. Now, these movements of materials don't require energy, but sometimes you require to expend energy because the materials that you're moving across the plasma membrane are more concentrated in one side than the other, and you're forcing to have a particular concentration. So this is known as active transport, in which you spend ATP, or adenosine triphosphate, and you will move the materials against a concentration gradient. So basically you move the materials from less to more. It's like if you're trying to pack a lot of people into when people trying to get into an elevator that's already packed, so you will require energy to enter into that elevator. So that's active transport. Now, bacteria has as well another mechanism of transport that is known as group translocation. And in this case, you will add materials or particles into this solute as the solute is passing through these transporter proteins. So we have, for instance, the use of phosphoenol pyruvate, or PEP, to add it into these molecules to prevent the molecules from escaping from the cells. Now, another structure inside the cells of bacteria is the cytoplasm. Now, cytoplasm will be the material or the substance that is found inside the plasma membrane. And inside the cytoplasm, the bacteria has ribosomes, and they have the nucleus, which is the area where you have the DNA. So this is the nucleus, and all of this is the cytoplasm. Inside the cytoplasm, there is this water that is called the cytosol, and then you have cytoskeletal proteins that are specific for the bacteria. Now, the prokaryotes have ribosomes because they produce proteins. Ribosomes are organelles that helps to produce proteins. And the prokaryotes, they have ribosomes that are known as 70S. 70S are made of, ribosomes are made of two subunits, 50S and 30S. These units, or these ribosomes, they have this sedimentation rate that is measured when you centrifuge proteins, ribosomes, in the centrifuge in a test tube. They tend to go to the bottom of the test tube, or into the sediment, at a particular rate that it is measured in Sedberg units. So these two subunits, the large subunit, which is 50S, and the small subunit, which is 30S, they join together, like in this case, small and large, they join together and they form a complete 70S. These are not additive. Don't think that because you have 50S and 30S, someone did wrong the arithmetic and didn't add into 80S. No, it's just that they have a different coefficient to go into the bottom of the test tube, but they don't add as a number. Along with this cytoplasm, the bacteria not only has these ribosomes, but they also have inclusions, which are like little pouches or internal structures that the bacteria uses to store energy most of the time. Some bacteria, they like phosphate groups, so they use metachromatic granules or bulletin to store phosphate. Some of them, they use polysaccharide granules in which they store glycogen. Some of them, they store fats, so these will be lipid inclusions. Some of them, they store sulfur because they use sulfur for making energy. Some of them, they have enzymes like ribulose 1,5-diphosphate carboxylase that they use to fix carbon dioxide to generate energy. In this case, this will be called carboxysomes. And some of them, they float within the environment and they use gas inside these vacuoles to float and will be aquatic environments. Don't think it's in the air. So then this will be called gas vacuoles. And some of them, they might have inorganic molecules like iron oxide. In this case, this will be used to destroy radicals of hydrogen peroxide that help for the bacteria to survive attack of oxygen radicals, and these will be called magnetosomes. And these magnetosomes can be seen really nicely in transmission electron micrograph. Like in this case, this is the middle of this rod, which has two flagella at the end. So this is polar Amphitrachus bacteria. And see, it has this row of magnetosomes where they store iron. Now, some bacterial structures that are important for certain bacteria, not all of them can produce bacteria, can produce this, is the endospores. So the endospores are like dormant cells. So these will be resistant forms of bacterial dormant cells that resist desiccation, heat, and harsh environments like chemicals, lack of nutrients. And two types of bacteria or species can form endospores, Bacillus and Clostridium. Bacteria that can form endospores undergo the process of sporulation on endospore formation. And when these endospores are released into the environment, they remain in the environment for years until they encounter the right conditions. And once they can encounter the right conditions, they can form a new bacteria that it is active. This is known as germination. And this is the return to what we call the vegetative state. So this is a Bacillus subtilis bacteria with this endospore. This is the dormant form of the bacteria. So this is the process of sporulation. So in order to form endospores, the bacteria has to duplicate its DNA. And once it duplicates its DNA, the bacteria will start encasing this DNA, wrapping it around the new bacterial DNA with plasma membrane first. And once this plasma membrane forms, you will have the formation of a spore septum around the isolated portion, which is known as forespore. And you will have two membranes instead of one, which happened in here. And then you will encase between the two membranes this peptidoglycan layer. And then you will have the release of this endospore, and the endospore will be free in the environment. So this is the sporulation process. And you will have an additional coat that it will be called the spore coat. So then those are the internal structures of bacterial cells. Now let's talk about eukaryotic cells. So eukaryotic cells, they have a nucleus, and this nucleus is located in here, and you will have two major types of eukaryotic cells. You can have plant cells or you can have animal cells. Plant cells, they have a cell wall, but you also have other eukaryotic cells like you have fungi, and then in this case, this diagram just shows you the plant cells and the animal cells. One major difference is that the plant cells, they have a cell wall, animal cells, they don't have a cell wall, and that plant cells, they have these organelles that are called the chloroplasts. And then animal cells, like in the case of sperm cells, they have a long extension that it is used for motility that is called as flagella. So this is a transmission electron micrograph showing you the differences between plant cells and animal cells. So here you have chloroplast, so this must be a plant cell. And well, in this case, no, this is an algae. This is not a plant cell. In algae, just like plant cells, they have this chloroplast. So they are capable of producing photosynthesis with this chloroplast algae, same as the plant cells. And this is an animal cell. So flagella and cilia can be found in eukaryotic cells, and it helps these cells to move. So flagella are, just as for bacteria, long extensions, and in the case of eukaryotes, these flagella, they have undulating movement or zigzag movement. This is euglena. This is an algae that has chloroplast and chlorophyll. Now, this is a protozoan. This is a eukaryotic cell, single cell organisms, and they move through the use of these cilia, not flagella. Cilia are shorter than flagella. And flagella, if you make a section of these, they have internally these microtubules in a 9-to-2 arrangement. So they have 2 central microtubules and they have 9 doubles around these central microtubules, and they are protected by a plasma membrane. But this is the common way then that eukaryotes move, through cilia, short extensions, or flagella. Now, the cell wall in eukaryotic cells that have a cell wall is much simpler than prokaryotic cell walls. And some of the eukaryotic cells that have a cell wall will be plant cells, algae, and fungi. The cell wall is mainly made of cellulose and chitin for algae and fungi. Yeast, they can have these sugars, glucan and mannan, in their cell wall. Now, some eukaryotes, just as bacteria, they have a coat outside the cells, which is glycocalyx. In the case of animal cells, the glycocalyx is made of carbohydrates with a negative charge that helps these cells to attach into the surroundings. And this glycocalyx, it also has these carbohydrates attached to proteins, in which you would call glycoproteins, or these carbohydrates attached to lipids, in which you would call it glycolipids. And the glycocalyx, it is protecting the plasma membrane of eukaryotes. Now, what are the components of the plasma membrane? So we saw then the external structures of the eukaryotes, which were flagella, cilia, and then we saw the cell wall in some cases, but we can have internal components to the cell wall of eukaryotes, like the plasma membrane. Now, the components will be phospholipid bilayer, peripheral proteins, integral proteins, transmembrane proteins, esterols, and glycocalyx, carbohydrates, in the case of some cells. Functions of the plasma membrane of cells in general will be act as a selectively permeable membrane and also allow the transport of molecules through simple diffusion, facilitative diffusion, osmosis, and active transport, just as for prokaryotic cells. Now, eukaryotic cells, they have a specific transport mechanism that is known as endocytosis. If the cell internalizes solid particles, this will be called phagocytosis. So the cells will extend false feet, pseudopods, that will engulf the particles. And if the cells bring in fluid materials with dissolved substances, this is known as pinocytosis. So this is exclusive of eukaryotes, endocytosis. Now the eukaryotes have in their cytoplasm cytosol, which is the fluid portion of the cytoplasm, and they have this cytoskeleton made of microfilaments microtubules, and intermediate filaments. So the microfilaments will be active. And then the movement of the cytoplasm to distribute the nutrients within the eukaryotes is known as cytoplasmic stream. And the cytoplasm of eukaryotes, they have a nucleus inside. And it also has enzymes, but these enzymes are found within the lysosomes instead of freely floating like in the case of prokaryotic cytoplasm. So for the nucleus, this is a transmission electron micrograph of the nucleus of a eukaryotic cell. It has a double nuclear membrane, which is this. And it has this mini organelle inside the nucleus, the eukaryotic cells that is known as nucleolus, where you produce the ribosomes. And then this nuclear membrane, it has these openings that you can see here. These are the nuclear pores. The nucleus is the area where you hold the DNA, either dispersed, that we know as euchromatin, or condensed in the form of chromosomes. And it's in this place, in the nucleolus, where you have the production of the ribosomes, the protein factories that makes the proteins. And then here you have the nuclear plasma membrane and the nuclear pores. And then the endoplasmic reticulum will be the next organelle that is nearby the nucleus. And this at least will be the rough endoplasmic reticulum that has attached to it ribosomes that make it look rough under the electron micrograph. So this is the rough endoplasmic reticulum with these dots that represent the ribosomes. And then the next organelle would be the smooth endoplasmic reticulum, or SER, where you don't have ribosomes and you have enzymes that help to synthesize lipids to form the plasma membrane, and also helps to detoxify the cells from alcohol or toxins. So this is the smooth ER in the electron microscope. So it doesn't have ribosomes. That's why it's called a smooth. Ribosomes are the sites of protein synthesis, and in the eukaryotes are larger and denser than prokaryotic ribosomes. So they are 80S. They have a 60S and a 40S subunits, and they can either be attached to the rough endoplasmic reticulum, or they can be free in the ribosomes. If they're free, in the cytoplasm, if they're floating in the cytoplasm, they will make proteins that will stay within the cells. But if they're membrane bound, like in the case of the rough endoplasmic reticulum, the ribosomes will make proteins that either goes to the plasma membrane or that they can be exported. And then in some organelles, like in the chloroplast and the mitochondria, you can have 70S ribosomes, similar to the 70S that you have in the prokaryotic cells. Now, once the proteins are produced in the rough endoplasmic reticulum, they will move to be packed and to be delivered by the Golgi complex into the right place. So the Golgi complex will be used to make these proteins available for the different areas. So it's like a UPS. It will pack and deliver these proteins. And is this series of sacs that can move the materials from one area into the other, and also the Golgi complex can form lysosomes, which has enzymes that can help to protect the cells or destroy the cells when the cells are worn out. Mitochondria is a sausage-like organelle, like this, that has two membranes, an inner membrane that folds in, that forms this cristae, where you have in the matrix inside this cristae enzymes that are important to generate ATP. And in this inner membrane, we have the electron transport chain as well that helps to generate most of the ATP in eukaryotic cells. And this mitochondria is the site then of cellular respiration, production of ATP. This is a transmission electron micrograph. And this mitochondria has its own DNA and it has its own ribosomes, 70S ribosome. In algae or in plants, you have this chloroplast that looks similar in shape to the mitochondria, and these are the place for photosynthesis. So you have these structures internally that are called the thylakoids that are held by these extensions that we know as granium. But inside these thylakoids, you have these enzymes that can convert 6 molecules of CO2 and 6 molecules of water, plus with the entry of this sunlight energy into them, you can form carbohydrates plus oxygen. So photosynthesis is important in plants and algae to help to maintain balance of excess of CO2 found in the environment. Now, small organelles membrane bound in the eukaryotes are lysosomes that has digestive enzymes, vacuoles that are also important for holding food for the cells and provide structural support. Now, lysosomes and vacuoles are formed within the Golgi complex, and peroxisomes will be these pouches that has enzymes like catalase enzyme that can break down H2O2, which is hydrogen peroxide, into water and oxygen to prevent oxygen radical damage. And also peroxisomes can oxidize fatty acids so that we can generate energy out of fats. And then the centrioles are organizing centers that helps to direct the microtubules to the centromeres when the cells are dividing in the chromosomes so that you can pull the chromosomes in opposite directions of the cells during cell division. So this is a summary of what we saw today. So we saw differences between prokaryotes and eukaryotes. Prokaryotes are simpler in general than eukaryotic cells. They are smaller with a size between 0.2 to 2 micrometers in diameter, while the eukaryotic cells, they have between 10 to 100 micrometers in diameter. Prokaryotes, they don't have a nucleus. Eukaryotes, they have a true nucleus. They don't have membrane-bound organelles. Prokaryotes, eukaryotes have membrane-bound organelles, and both can have glycocalyx. Some eukaryotes can have a cell wall, like in the case of plants, algae, and fungi. In the case of prokaryotes, most of them, they have cell wall, with the exception of mycoplasma. And in the cytoplasm, there is no cytoplasmic streaming for prokaryotes, while in eukaryotes, they have cytoplasmic streaming. Plasma membrane, in prokaryotes, they don't have carbohydrates, and they don't have esterols, with the exception of mycoplasma. While in eukaryotes, we have carbohydrates that serve as receptors, and we have sterols. DNA typically is circular in prokaryotes with only one chromosome, and they don't have the DNA of the chromosome, they don't have histones. While in eukaryotes, we have multiple chromosomes with histones, and the chromosomes are linear. Cell division prokaryotes is through binary fission, eukaryotes is through mitosis. Sexual recombination in prokaryotes is through the transfer of plasmid DNA, if it happens, and then in eukaryotes, it involves meiosis. So this is just to show you the difference between eukaryotes and prokaryotes in terms of size. So these are eukaryotic cells. This is the nucleus, this is the cytoplasm, and all of this is prokaryotic cells infecting these eukaryotic cells. And this is a view with light microscopy. So even with light microscope, we can see the size difference between eukaryotic and prokaryotic cells. So some highlights of this chapter will be that bacteria have unique cell structures that are not found in eukaryotic cells that can be target for antibiotics, immunity, and phage infection. Now, bacteria and archaea, although they have similarities and they have specialized structures like flagella, endospores, and pili, These structures confer critical capabilities that helps them to attach to surfaces, helps them to survive harsh environments, and they help them to actually act like antigenic molecules and also helps them to detect them or to classify them as subspecies. And last highlight is while microscopic eukaryotes like fungi, protozoa, and algae can carry out some of the same processes as bacteria, many cellular properties are fundamentally different between eukaryotes and prokaryotes. So this is all for this chapter. I will see you in the next. Bye-bye.